First, we're going to put some strawberries without the stems and leaves in a Ziploc bag and smash them. Then, add 20 ml of extraction liquid, which is a mixture of distilled water, salt and dishwashing liquid, into the crushed strawberries and squeeze them again.
Next, we strain the strawberry mixture into a beaker to get rid of all the pulp. This might take a while.
One eternity later.
The filtered liquid is put into a 50 ml tube. Then, cold rubbing ethanol was poured down the side of the tube to make 2 layers.
I wonder what happens when we dip the glass rod in.
That's the extracted DNA. Let's put it in a sample tube with 0.5 ml of glycerin. That's it for DNA extraction. Now let's move on to gel electrophoresis.
First, we prepare the wooden comb by sandwiching 4 evenly spaced coffee stirrers between 2 big ice cream sticks and taping them together. This will be used to create wells for our samples later.
Next, we prepare the gelatin. Weigh 2 grams of gelatin powder and add 200 ml of 0.2% sodium bicarbonate buffer. Then, we heat the solution in the microwave.
After the solution is warm to touch, and just pour it approximately 0.5 cm above the tip, like this. Just pour a little, and then we're gonna put it into the freezer.
After the gel has solidified, we cut out the ends of the gel and remove them to make room for the electrodes. Next, more buffer is added until the electrodes and teeth are submerged. Then the wooden comb is carefully removed to allow the buffer to fill up the wells.
Now, we load each well with 5 ml of DNA sample, methylene blue, red food coloring, It's okay guys, mistakes happen. And last but not least, we add the green food coloring. Then, connect the electrodes to a power supply and leave it for about 15 to 20 minutes.
After the power supply has been turned off and the electrodes removed, a few drops of methylene blue solution is added onto the gel and gently spread. This is to ensure even staining for the DNA. Then we remove the gel from the container to observe.
Here's what our gel ended up looking like. Since there were a few errors in our experiment, the results were not that accurate. Let's discuss. First, the red and green dye moved towards the positive electrode because they are negatively charged. The green dye split into 2 colors because the molecules of both pigments are different sizes. The methylene blue moved towards the negative electrode because it is positively charged. That's why it moves in the opposite direction of the dyes. And theoretically, DNA or nucleic acid should move towards the positive electrode because it is negatively charged due to the phosphate group. And that concludes our experiment. Thank you!